Hi, I'm Charlie Stagg and I work at the Wynn in Oxford, where I head a research group interested in human motor plasticity. One of the things we've become very interested in is neural dynamics and their involvement in motor learning. And so I'm going to use that as a case study today to talk about a couple of studies highlighting how we go about using MR to inform us about the effects of electrical stimulation and, in particular in this case, alternating current stimulation. So I know many of you may be less familiar with neural dynamics. So when we ask people to move and we record their brain activity using MEG or EEG, we see an absolute characteristic pattern of activity, and particular a decrease in activity at 20 Hertz in the beta band as people prepare and execute their movement. That's an absolutely classic finding, and we've known about it for decades. But what we don't really know is what role that has in motor learning, if any. So in my group, one of the things we've been interested in is following that up. And so Katerina Zich did a study where she asked people to learn a motor learning task where they had a 4-script transducer, which they had to squeeze to move a cursor to a series of gates and back to the home condition. They did that in the scanner in the MEG for about 20 minutes. And as you might imagine, over that time, they got faster and more accurate at the task. What we then went on to do is look at the neural data and look at the brain states associated with that learning. We used HMM and identified a brain state that had a peak of activity at 20 Hertz. We then looked at what happened to that during movement and showed that, analogous to the time frequency data, that showed a decrease in activity in movement preparation and execution. Interestingly, over the time of learning, that decrease in beta became stronger as people learnt, and on a subject-by-subject basis, the degree to which that changed correlated very nicely with their improvements in both movement time and accuracy. So it seems that 20 Hertz stimulation or 20 Hertz activity might be very important for learning. So we then were interested in 20 Hertz tax and wanted to see if there was evidence that this might have a behavioural effect. And there is some. So this was a paper from a few years ago where the authors asked participants to do a finger tapping task at an interval of 900 milliseconds. When they gave them 20 Hertz tax, what they saw was a significant slowing after stimulation compared with sham. But the results are not consistent across the literature, and it's not clear why some studies do see effects and others don't. And one of the reasons for that may be that we really don't understand the neural mechanisms of stimulation, so it's difficult to predict who might respond and when. One of the ways of starting to think about what tax might do to the brain is to start to think about the communication through coherence hypothesis. So in this cartoon model, we have 3 brain regions. Area A is functionally connected with Area C in a positive way because the underlying activity in Area A is in phase with that in Area C. Area B is either inhibitory or not connected to Area C because its underlying activity is either out of phase or at a different frequency to that occurring in region C. So if we stimulate Area B, for example, with tags, perhaps what we're doing is, by applying an exogenous rhythm, moving the ongoing activity either in frequency or phase within Area B, thereby potentially lining it up with Area C, changing the connectivity and increasing connectivity between those brain regions, and hopefully therefore improving behaviour. But that hypothesis raises 2 very big questions that need answering. One, is there any evidence for any physiological changes within Area B in this example that might explain how tax is able to modulate the phase and the frequency of ongoing activity? And is there any evidence that modulating one region with tags changes connectivity within the network? So in terms of the physiological changes, for us, the logical way to start looking at this was to use MR spectroscopy.
So for those of you who aren't familiar with this technique, what we do is we put people in our 7 Tesla magnet in Oxford and we record a signal from a 2 by 2 by 2 centimetre voxel located within the primary motor cortex. If we plot the signal coming back from that area as a function of its frequency, what we see is a characteristic spectrum which allows us to quantify the concentration of a number of neurochemicals, including glutamate and GABA, the major inhibitory neurotransmitter. And so what Magda Novak did, who was a student in my group at the time, was use MR spectroscopy to interrogate what the neural effects of tax might be. So what she did was take a group of healthy people and ask them to perform a motor task while she recorded EEG. And that allowed her to identify the individual frequency, peak frequency, within the beta band for each subject. What she then did was pop people in the scanner and recorded MR spectroscopy data from the M1 during tax stimulation at 20 Hertz compared with a sham control. These are the group level results. As you can see, there is a significant decrease in GABA on average during stimulation, but it's not particularly substantial, and it disappears after stimulation. Similarly, there's an increase in glutamate concentration in this region during stimulation, but that disappears after stimulation. But what we were really interested in wasn't group mean changes, but was rather whether or not GABA concentrations might tell us about the physiology of the changes of tax. So you might imagine that some people's individual beta frequency is higher than 20 Hertz, and what 20 Hertz tax might be doing is slowing it down. Some people's may be slower than 20 Hertz, and therefore what the tax is doing is speeding them up. So can we look at the deviation from 20 Hertz of that peak frequency and see if there's any changes in either glutamate or GABA that relate to how much individuals are having to change their frequency to match the tax? And what we see is a linear relationship between GABA change and that difference in frequencies. And that's specific to GABA and it's specific to beta stimulation. So that's not, of course, giving conclusive proof that inhibition is important in changing frequencies to allow these regions to align, but it gives us a first idea that perhaps that might be a mechanism by which the cortex is able to be entrained by the alternating current stimulation. The second question that we wanted to ask was about network connectivity. Specifically, if we stimulate one region of a network, can we see changes in the connectivity of its functional network? There are a number of ways that you might think about doing that. We work at the WIN, and therefore we used an ICA approach as illustrated in Melodic, where we looked at the motor network and we had the default motors of control. And we took a group of healthy subjects and put them in our 3 Tesla scanner in Oxford and gave them 3 runs of resting state activity, fMRI, where we did either 20 Hertz stimulation, 5 Hertz stimulation as an active control, or sham. And the order of those was counterbalanced across the group. And what we saw was that beta frequency tax, so 20 Hertz tax to primary motor cortex, had no effect on the overall connectivity within the motor network. Similarly, we can look at connectivity between motor cortices. So we know that most of the connectivity in motor cortex is with the other motor cortex, so we can look at that connection. And similarly to the overall network strength, we saw no change in overall connectivity of the primary motor cortex. Previously, our group and others have shown, however, that there is a significant relationship, as you might imagine, between overall connectivity in the motor network and connectivity between the 2 major network nodes. And again, here in sham stimulation, we replicated that relationship. We maintained that relationship with 5 Hertz stimulation, but it disappeared with 20 Hertz stimulation. We were interested as to the reasons that happened, and so we set up a little toy model that allowed us to explore this. And what this showed was exactly what we'd seen empirically, that this relationship between motor network connectivity and M1 connectivity was maintained with sham and 5 Hertz stimulation, but disappeared at 20 Hertz.
Now, models like this can be shown to show anything. So what we wanted to do was to derive a hypothesis from the model that we could then go back and test in our empirical data. And one of the things our model told us was that the degree to which motor cortex activity or connectivity to the other motor cortex changed should be related to how connected they were in the first place. And that's something we could go back and test in our empirical data, and exactly what we found. So I'm going to finish there. I hope I've shown you a couple of studies that give you a little bit of an idea of the kind of things we can start to do with MR to look at the effects of alternating current stimulation. We've used spectroscopy to interrogate the neurochemical changes due to TACs. And we've used resting state fMRI to look at network level connectivity changes. And the reason for doing this is if we can understand the underlying physiology, then we can try and optimize the approaches. And this gives us a new avenue to develop potential therapeutic approaches and neuroscientific approaches. So I'm going to finish there. Thank you very much for your attention, and thank you to my group and to my collaborators in Oxford and elsewhere for all their hard work. Thank you very much.
Hello, my name is Dave Sharp. I'm ringing in from London in the UK. I'm going to be talking today about the use of transcranial electrical stimulation with a simultaneous functional MRI experiment. So my group's been interested for a while in the use of network diagnostics, so defining the structure and function of brain networks. So for example, the salience network or the default mode network, and looking at whether that influences the response to treatments. So for example, drug treatment or, as we're discussing today, non-invasive brain stimulation. So my group's been particularly interested in problems that arise after traumatic brain injury, and often patients suffer with difficulties with cognitive control. So these are the set of functions that allow you to control your behavior. And one of the critical functions is response inhibition. So this is the ability to inhibit or stop unwanted actions. And one of the key ways that this is investigated is using a task called the stop signal task, illustrated here. So it's pretty simple. You generally see an arrow pointing left or right. You press left or right in response to the arrow. And then on some of the trials, here 20% of the trials, you'll see a red dot. And in that situation, you have to inhibit your response. Now the red dot appears at some point after the arrow, and the timing of the red dot in relation to the arrow changes to make the task harder or easier. And by titrating the timing of the red dot, you can basically measure an individual's ability to inhibit their actions. So we know a lot about the activity of the brain during performance of this particular task. So this is the network of brain regions seen during stop signal task performance. It's been studied in a large number of investigations. And what you can see in red are some of the areas activated during successful stopping. So when you successfully inhibit your response, you'll see activation of large parts of the right frontal lobe, in particular parts of the right inferior frontal gyrus and right anterior insula, and also parts of the anterior cingulate cortex. And these are regions that form part of the salience network that responds when behaviorally salient events occur, and often you need to reorientate attention. In addition, at the same time as this activation, you'll see deactivation of the default mode network. So you see this balance of activation within the salience network and executive control regions, and then deactivation of the default mode network. So we know that the connections of the salience network are important for its function. And one particular tract, the frontal Aslan tract, connects parts of the inferior frontal gyrus, and also beneath it the anterior insula, to the pre-supplementary motor area and the anterior cingulate cortex. And when this is damaged, it produces problems with response inhibition. So we see that in traumatic brain injury, and also abnormal default mode network activation and abnormal functional connectivity between these two networks. So damage to the structure of the salience network is important in determining the behavior that's associated with it. So we were interested in working out whether or not we could use transcranial direct current stimulation, TDCS, to stimulate the salience network and to potentially improve cognitive control. And we specifically wanted to test whether we could enhance response inhibition as measured by the stop signal task, both in healthy subjects and in patients following traumatic brain injury. We had a setup where we could do simultaneous TDCS in the scanner. So we recorded functional MRI at the same time as providing TDCS stimulation. And then we could look at whether stimulation modulated brain activation or deactivation during performance of this task. And also we wanted to specifically test whether the response of this was influenced by the structure of the salience network, and in particular its structural connectivity, so the structure of the white matter tracts within the network. So we used, as I said, the stop signal task. We had a setup where we were using anodal or cathodal stimulation and also sham stimulation. And this is the electric field map of the stimulation location. So we were trying to stimulate the inferior frontal gyrus and also the underlying anterior insula. And you can see stimulation effects within the inferior frontal gyrus here, and also underneath within the insula. And also I've overlain the tract here, the frontal aslant tract, and we can see that the location of stimulation is very close to the location of this particular tract. So the first result that came from our healthy control study was that stimulation of the inferior frontal gyrus and the anterior insula did produce some modulation of activity on the stop signal task. So the stop signal reaction time, the SSRT, is the key behavioural measure, and low SSRT is a measure of improved response inhibition. And we can see here that anodal TDCS produces a small but significant improvement in response inhibition on this task. Now we wanted to look at the structure of the network, and so as I mentioned, we were particularly interested in the connectivity of the salience network, and so we measured the structure of this particular tract using diffusion tensor imaging and measuring the fractional anisotropy of this tract. And what we can see is in the anodal situation, there was a big effect of the structure of this tract on the response to TDCS. So specifically, if you had high FA within this tract, your improvement on the SSRT was very significantly greater than if you had low fractional anisotropy. So essentially, the structure of the tract, particularly in the context of anodal stimulation, influenced your response. That was the behavioral response. And the same was true on the physiological response. So as I mentioned, we had functional MRI at the same time. And here we can see activation of the anterior insula during successful stopping. And in the situation where the connectivity of the network was high, showed an increase with TDCS. So the physiological and behavioural effects of this stimulation was dependent on the structural connectivity of the tract. So as I mentioned, the clinical focus of the group has been around traumatic brain injury. So these are injuries often caused by road traffic accidents, but sometimes by other situations like sporting injuries. And the reason for particularly looking at this in this context is because TBI produces axonal injury, so damage to the axonal connections within the brain. And so we think that this is important in determining the kind of treatment that we want to give people and might influence the effects of non-invasive brain stimulation, as we're discussing today. So we can use diffusion tensor imaging to measure the white matter integrity within the brain. And here we can see the structural connectivity of the tract that I've been talking about, the frontal aslant tract, in our patients and in our controls. So we see a reduction in FA quite markedly within the patient group, which is what we'd expect because of the presence of axonal injury in a proportion of the patients. So there's damage to this this network that we're trying to stimulate. So the question is, does that influence the effects of stimulation there? And the answer is yes, it does, in a similar way to in the control subjects. So what we see plotted here on the x-axis is the fractional anisotropy. Reduced fractional anisotropy is worse, so indicative of damage. The dark grey dots are the TBI patients, and we can see that as the TBI patients in general show reductions in FA, as I've told you. But then the change in SST produced by stimulation is correlated with the fractional anisotropy within the network. So as the structural connectivity it becomes more impaired within the network, the effect of SST becomes less. So here, the patient or the controls with very good structural connectivity within that network showing a big response to TDCS, and here are in general the patients with damaged networks showing no benefit and potentially a impairment of response. So finally, there was also a physiological impact in the patients of this. So we can see a marker of poor performance in this group is the failure to deactivate the default mode network, here seen as the posterior cingulate cortex. And damage within the Salience network is correlated quite strongly with this failure to deactivate the DMN. This is a slightly complicated figure from our paper, but essentially what it shows is that in general, in areas that are activated during TDCS, there wasn't much change in the physiological effects. But what we saw was a normalization of the deactivation in the TBI population, and particularly those patients that were impaired on the task. So there seemed to be a physiological effect of stimulation that was seen within this connected network, with a somewhat normalization of the pattern of response seen there. So just to finalize things, so in conclusion, what we've shown is the white matter structure of a stimulated brain network influences the behavioral response to that stimulation. And we've seen that in the context of the Salience network, and how connectivity of the Salience network, particularly the frontal aslant tract, influences the behavioral and physiological effects of TDCS stimulation on executive function. And we see that both in the context of healthy subjects and also patients with cognitive impairments after traumatic brain injury. And so I think what this suggests from a clinical perspective is that it really suggests we need a personalized approach to non-invasive brain stimulation, which factors in some measurements or some information about the structure of the networks that we want to stimulate. And this is likely to be necessary if we're able to produce clinically important effects. I'd like to acknowledge the funding for this work, particularly the Medical Research Council in the UK and the Wellcome Trust, and also my colleagues, in particular Dr. Lucia Lee, whose PhD project was involved in most of the work here, and also Inez Violante, who did much of the work supporting Lucia. Thank you very much.
Hello, my name is Marom Bikson, and I'll be talking about using functional imaging as well as EEG in combination with head modeling in order to make transcranial electrical stimulation, or TS, more specific or more individualized. So these are my disclosures, as well as the source of support. And on my slides, as well as the references I'll be talking about, are at this, can be found at this Twitter account. So just briefly, transcranial direct current stimulation, or TDCS, is a wearable non-invasive neuromodulation technique. And it's unique from other forms of brain stimulation in that it doesn't use pulses. It uses a continuous slow DC waveform. And that DC waveform is low intensity. It's about what you get out of a 9-volt battery, but it's current controlled to a level of 1 or 2 milliamp. And it's applied for typically tens of minutes, so 10 or 20 minutes. And the electrodes can be positioned on the head in different ways. So what's shown here is the M1 motor SO supraorbital montage. And I'll be discussing how to combine or leverage imaging with TDCS and also a related technique called transcranial alternating current stimulation. Now, we recently published a review that outlined all the different types of considerations that would go into an experimental or interventional attempt to combine imaging with TDCS. And I'm not going to unpack all of those here. I think you'll hear about some of those in the other talks. I'm going to be focusing specifically on how to use computational modeling of brain current flow in the context of imaging and TDCS. And in this paper, we do talk about some critical caveats in how you combine TDCS and imaging, but I'll be focusing on just this one issue of using current flow models. So some of my work here began in 2014 in collaboration with Andre Antal and Walter Paulus and others in Göttingen, Germany, where they did a trial where they conducted functional MRI, standard bold imaging in dead subjects. So these were people who had recently passed away, and they were wheeled into a scanner, and they were imaged during the application of transcranial direct current stimulation. Again, using these are standard sequences. And you can see below some of the results that they got. They got very robust, reproducible, and polarity specific. So you can either have the anode electrode here and the cathode here, or the cathode here, or the anode here. That's what it means to switch polarity in the context of transcranial direct current stimulation. So they saw montage specific, this is the M1SO montage, and polarity specific bold responses. And again, these are subjects who were deceased. And so we worked with them, and what we did is we related the signals that they were detecting during application of transcranial direct current stimulation with our computational predictions of the current flow through the head during transcranial direct current stimulation. And what we found that these were highly correlated, and especially note the fact that when you stimulate the head, a lot of the current concentrates in the cerebral spinal fluid, not in the brain. And that is actually where they were seeing most of their signal. So let me take a step back and describe how these models are generated. The basic pipeline for generating MRI-derived, or as we call them, gyri-precise models, was something we proposed in 2009. And it starts with high-resolution anatomical scans. These are T1s, typically 1 millimeter in all axis. And once you have these scans, you then segment the tissue into the relevant masks. And because we want to simulate current flow through the entire head, it's not just the brain or separating the gray and the white matter that matters to us. It's critical to segment the skull, the CSF, and the skin, and other relevant structures correctly. And so the pipeline that we established in 2009 really set the stage for these gyri-precise or individual models. And when we do these models, we can position the electrodes on the models analogous to the way they would be placed in the person. So in this case, this is the M1SO montage. And the models predict the current flow passage through the head, through the skull, through the CSF, through the brain, through the deeper brain regions. But often when we're showing the results of these simulations, we're focusing on the cortex or the surface of the head, so how current is penetrating through it. And we show this in terms of a false color map, typically where hotter colors are regions which get more current flow, and black or cooler colors are regions that don't get more current flow. And this represents the electric field or the current density in each voxel of the brain. So this is what those results look like. This is the prediction of the brain current flow during the M1SO montage. And you can see what we showed in this paper is that when you take two large electrodes and you place them on opposite sides of the head, the current doesn't just flow in, let's say, the motor strip, but it diffuses broadly through the head. And you can also see, you see these little clusters. These really represent idiosyncratic anatomy of the cortex and the folding. And so current flow is diffuse. It's also deep. You can see in the cross section, and it's under both electrodes. And so these models predict the current flow pattern through the head. We can vary the montages that we simulate to ask how does electrode position change where the current goes. one of the ideas we also developed in the same paper was something called the 4x1 high definition TDCS montage. Here you can see the electrodes are smaller. There are also more of them, and they're arranged in such a way that you have a center polarity, and the 4 electrodes on the outside have the opposite polarity. And what we predicted in this paper, again, showing you a current flow simulation, but focusing here on the cortex, is that when you stimulate in this way, you produce a current flow pattern that is much more targeted. So using the 4x1 high definition TDCS montage, you can be non-invasive, electrical, and targeted. And that was a major prediction from this paper that has since been validated. So I'm just showing you how the models can be used to compare a variety of different forms of montages, both pad montages and a whole variety of different types of high definition montages. But in any case, the product of these models is always a false color map showing how much intensity each brain region is receiving. So that's what's being shown here in a slightly different color map here, where blue, the cooler, is no current density, and red is the most current density. And so you can see you have the called the bold signal that was picked up from these subjects. Again, these are deceased subjects. And then you have the current density predictions. And you can see that there is a strong match between them. You can see that just by eye. We can also demonstrate that statistically, meaning that the scanner is picking up not brain activity, as was maybe obvious, but it's actually picking up some sort of hallmark of the current flow through the head, which doesn't necessarily have to be the brain. It can be the CSF as well. And so really what we're showing is that a conventional imaging technique, right, just standard bold sequences, are highly sensitive to current flow. And the reason this is, as we showed in this paper, is that the current flow produces a magnetic field, and that magnetic field then interacts with the scanner's own imaging sequences. And so the result is what looks like a bold response, but is actually a signal artifact. And that was, again, unpacked in this paper. And so we show in this paper that when you do MRI studies with online TDCS, you need to be extremely careful. You need to be extremely careful because you will pick up an artifact, and that artifact, by the way, will be montage specific, right? So a lot of the control experiments you might do to control for that artifact are not going to work. We also showed, though, in this paper that transcranial alternating current stimulation, or TACS, was artifact free, at least the way the scanning was done in this case, and we explained why that was. Now, since that paper was published, we and others have actually focused on not just using standard imaging sequences, but developing optimized imaging sequences in order to more, with higher sensitivity, detect the current flow flowing through the head. So then again, this is using the MRI not to measure brain activity, but to actually map current flow through the head, essentially validating these models. And there's also been work by us working with Danny Wang and his colleagues at UCLA, as well as Duke Shireen, who's in my institution at the City College of New York, where the goal there is to say, well, we know we have these artifacts during TDCS, but because they are essentially a known quantity, can we correct for them? And can we, in fact, with correct and careful image processing, image brain activity online during TDCS? So I mentioned this review that we published, which starts to unpack all of these issues. Now, generally speaking, when you talk about how you can combine imaging and stimulation in very powerful ways, well, for one thing, functional MRI is often used to identify the target of brain stimulation. So separate from stimulation, subject would be scanned during a task, and you might identify the target that way. How do you get to that target? You now need these anatomical MRI-derived models that will tell you where to position the electrodes in order to get to that target. As I just mentioned, we can actually use the MRI to validate the same current flow models. And then finally, as we unpack in this paper, there's a possibility to combine imaging and current flow modeling to do analysis of brain effects of stimulation in a much more nuanced way. For example, we can apply stimulation, we can predict which brain regions get the most current flow, and we can use that in our analysis let's say, fMRI imaging response of how TDCS affects the brain. And as far as making these modeling tools available, there's various commercial and open source possibilities out there. This is one that we have released and continue to update. It's called Roast. It can be downloaded for free, and it's relatively simple to use. It's free, but it works within MATLAB at the website that's indicated here. Now, finally, I'm just going to briefly mention that a lot of the issues that we mentioned with fMRI and other forms of MRI-based imaging apply equally for other forms of imaging such as EEG. And as summarized in these references here, this is in different ways. One of them is that we can actually use the EEG to guide how we position the electrodes in order to stimulate a specific target. This is a long-standing idea. It's something called reciprocity. It basically says that if a brain region is active and we pick that up as an electrical signal on the scalp, we can take that electrical signal on the scalp and we can invert it in such a way that we then drive current back to that very same region. And what's peculiar about this is you don't actually need to localize the source to do it. And so we've published 2 papers on that are indicated here. One does not require MRI, and it's relatively straightforward. The other one is a closed form solution. But what we demonstrate in this paper, essentially solving reciprocity, is that reciprocity requires individual MRI. Now, there are also, as I mentioned in the next bullet point, serious concerns with online EEG and TDCS, as well as online EEG and TACS. These things interact in complicated ways. We call them inherent physiological artifacts because it doesn't matter what kind of equipment you have. The quality of your stimulator or the quality of your EEG equipment obviously matters, but even with ideal performance, these artifacts are inherent. And so therefore, they will always be there during online TDCS, TACS, and EEG. And therefore, for any form of online analysis, they must be directly recognized and they must be addressed. And finally, there are experimental design related issues, just like they are when combining TDCS and imaging. So thank you for your attention to this talk.
Hello, my name is Ekhtiari from Tulsa, Oklahoma, Laureate Institute for Brain Research. I'm really happy that we have this symposium at OHBM 2020. I was really hopeful that we will have it at kind of in a physical presence in Montreal, but it seems that we are going to do that online. So we have a specific symposium on symbiosis of fMRI and transcranial electrical stimulation. And me and my kind of colleagues that are going to present you some ideas about the interesting potentials with the combination of these 2 areas are hopeful to be able to kind of motivate you to join us in these really interesting areas of research in basically human brain mapping. So just to give you an introduction, and we talk about transcranial electrical stimulation, we are talking about delivering a really little amount of current for something like kind of few milliamps in the direct current or alternating current. And so we can have transcranial direct current stimulation, transcranial alternating current stimulation, or sometimes random noise stimulation. When we talk about symbiosis of fMRI and transcranial electrical stimulation, there are basically some major challenges or major potentials in the field, I should say. At least I'm going to target some of the kind of major challenges or major potentials, 3 major items I'm going to cover today. So hopefully you will have an idea about what are the interesting potentials and challenges in the field. When we talk about using fMRI in the kind of the TES or TDCS trial, we are talking, for most of the studies, we have a mechanistic kind of question in terms of, okay, if you do fMRI before and after, how can we see what is happening inside the brain by using brain stimulation? So that is a mechanistic question that we have. But there are studies that people might also kind of try to use fMRI as a predictor or as a response that is going to be predicted. So there are some prediction studies that people try to see if they can predict what is going to happen with basically with TDCS or TACS using fMRI. And also we have the 3rd group of the studies that people try to inform the electrode montages and the way that they are going to stimulate based on the fMRI data. So we have another group of studies trying to use fMRI and that way. So we basically have three major types of integration of TES and fMRI. We have recently done a systematic review with a brilliant PhD student, Peyman. Peyman has done a really nice job on kind of evaluating all the published studies that we have in the field. So we had collected 138 original papers that composed of 220 different experiments of combining basically TES and fMRI. As you can see here, we have 202 studies that are basically mechanistic in sense of using fMRI to see how TES or TDCS is changing the activation inside the brain. 163 with direct current and 39 with alternating current stimulation, okay? Including the studies that we have done in terms of showing that, okay, how TDCS could modulate brain activations using fMRI before and after. So I'm not going to go to the details about that, but these are basically the mechanistic studies. But when you talk about these mechanistic studies, you really like to know, okay, what is the conclusion of these mechanistic studies? When you target a specific area in the brain, how basically TDCS is changing the activation inside the brain. Two postdocs from Michael Nichel lab, we have done a collaboration to see, okay, if we go to all those studies that I told you before in the systematic review, what are the kind of the changes that we have inside the brain? As you can see here, when we target a specific area in the brain, there are people reported that there are activations or changes in the fMRI activations or connectivities within different areas in the brain. Of course, I mean, there are reports that when we kind of stimulate a specific area in the, let's say, prefrontal cortex, we have changes in the prefrontal cortex activations or fMRI signal. But it's interesting that especially for the prefrontal cortex, we can see that there could be kind of changes in the activations in other areas in the brain. And that would make things really interesting in terms of, okay, when we talk about brain stimulation, especially when we talk about prefrontal brain stimulation, we are also talking about modulating subcortical areas based on the connectivities that we have with these areas. Those who are interested to have more details about the systematic review and kind of the results, we have recently put that in medRxiv so people can go inside and see the details about the results that we have. But based on the number of the studies that I told you, you might say that, okay, it might be good to do a meta-analysis to have a better sense about, okay, how direct current stimulation on a specific brain area would change the activation inside the brain. So we had 163 mechanistic studies with TDCS, and 71 of them are on basically sensory motor area. So they put the electrode over the sensory motor area. So that is a kind of reasonable number to be able to do a meta-analysis using different meta-analysis way that we have with neuroimaging and fMRI data. But when you go to the details of these studies, you can see that there is a huge variation in terms of the methods that people have tried to use. People have used task-based, resting state, different intensities of current stimulation, different variations of duration, different methods of applying the current. So it's really hard to find two single studies that they replicated the results, because I mean they have used really different variations. So it's really hard to do any really high quality kind of meta-analysis or any sort of conclusion in the field how TDCS is changing the activation in the brain, even on the kind of montages or kind of targets that we know that we have many studies inside. So we realized that we need to have a level of both harmonization, also transparency, because it seems that people are not sometimes reporting all the details that we need to be able to conclude. So we moved forward and we made an international network of TES fMRI, basically labs that are doing TES fMRI. We sent an invitation to a group of colleagues, and we ended up having over 50 labs across the board joining to the network. And we are running webinars, kind of different sorts of meetings together. So it seems that we are pretty much in a good shape in terms of establishing collaboration on that level. So if you are interested, please just send us an email or find us on Twitter, and we definitely would be happy to kind of help you if you want to set up a study in the field in a kind of a scientific rigorous way. The second issue that we have in the field is we are facing with a kind of huge level of inter-individual variation in response to kind of electrical stimulation inside the brain. We know that when we talk about kind of head modelings and the level of current that we have inside the brain, as you can see here, we can have a variation in terms of how much current we can have in different parts of the brain. So we have different dosage of current in different individuals. This is one of the studies that we have done here with 65 subjects. As you can see here, we have a huge variation in electrical current distribution inside the brain. So the question is, okay, how we can inform fMRI based on the electrical kind of dosage in different parts of the brain, and how we can integrate these two sort of kind of brain mapping modalities together. So we can have integration in different levels. We can have integration in the whole brain level. So we can, within each voxel, for each voxel, we can integrate the data. So there are challenges for that, but that is one thing that we can do. We can also do what we call region-based integration within specific ROIs. We can think about how to have a specific integration within the clusters of activation in the fMRI data. Or we can even think about network level integration of the data that we have from the computational head model in terms of the dosage of electricity that we have in different parts of the brain in combination with what we have in the larger scale network or smaller scale networks in the fMRI. Just to give you an idea about one of the kind of potential things that we can do, just two examples, as you can see here, with two different montages, F4, F3, F4, FP1. So we explored how we might be able to integrate the computational head modeling in the subject level with the fMRI data, with also kind of the network-based level data. Another really brilliant PhD student, Ghazale, has done a really nice job putting these things together in both larger scale networks and also in the nodes of different networks, and kind of explored how we can do the integration and what are the networks that would be affected more or less with the stimulation, different stimulation protocols. Those who are interested, Ghazale is going to present her results in one of the kind of poster session, poster number 17, so you can go and see the details that she is going to provide. If you want to have more details about some other case studies that we have done on integration fMRI and computational head models, you can read another paper by Zainab, one of the PhD students from Marom Bikson team. She has done a really nice job in putting together details about that. So you can read all those details on that publication, Human Brain Mapping. The third issue is also really important, is about how we can optimize the stimulation protocols for each individual subject. And that's another challenge that we have in the field. So we are running right now a trial, what we call a frontoparietal synchronization. So we try to increase the connectivity within the executive control network by using alternating current stimulation, synchronized alternating current stimulation between nodes inside that specific network in the frontal and parietal areas. But one of the challenges that we have is about what would be the optimum frequency, or even sometimes what would be the optimum phase difference between these two basically electrical stimulation that we are applying in these two areas. As you can imagine, there would be a kind of large parameter space when we talk about different phase differences, different kind of frequencies, and that would make things really complex in terms of how to find the optimum kind of stimulation protocol for individual subject to be able to make the highest level of connectivity between these areas. We recently developed a new setting on being able to do concurrent electrical current stimulation inside the scanner. And we have done different testing recently. It seems that I was really skeptical in the first days, to be honest, but it seems the result is much better than what we have been expecting before. So right now we are moving forward to do what we call a closed loop TACS fMRI. So what we do, we basically measure the connectivity between frontoparietal nodes, and then we stimulate, and then we measure the connectivity again to see how we can find the optimum stimulation protocol to do the stimulation. And in this figure that is provided by Lorenzo, I mean, people in the Imperial College, they have done a really nice job in that field. Some really kind of preliminary studies, but it seems that field is expanding. They are using kind of large pads of electrodes. We have recently developed really small electrodes to be able to do the same, let's say, sort of focus stimulation inside the brain. So that is another development that we are working on that. Another brilliant PhD students, Benny from Oklahoma University, is kind of doing his thesis project on that. We have collected one subject right before the pandemic, so the center is shut down right now, but we are just getting ready probably in the next probably in the next 2-3 weeks to be able to be back and run this experiment of closed loop resting state and transcranial electrical stimulation together. But as I told you, there are some challenges in terms of doing concurrent TS-fMRI. There are also really great potentials for that. So we made a specific collaborative network of labs that are interested to work on concurrent TS-fMRI. And we developed a methodological checklist in terms of the details that you need to pay attention to when you want to run a trial and that quality. And those who are interested, they can find the details about the checklist on OSF. You can just search for that. There are some details inside, and those who are interested to collaborate, they can just send us an email, and we will definitely be happy to help for that. So just to conclude, it seems that there are really interesting, exciting potentials for combining transcranial electrical stimulation and an fMRI, and there are definitely lots of challenges that we need to kind of handle in the future. I think that we're still in the kind of first step to have a better idea about how polarity, intensity, and duration and montage of electrical stimulation could basically change what we have inside the brain. I'm going to refer you to a paper that we published in Neuron about those challenges. And I think that the paper is still really helpful for those who are interested to start to have a better idea about potentials in the field. Okay, so I'm out of the time, so I need to kind of thank my colleagues and collaborators, especially Michael Nietzsche, Marom Bikson, Vincent Walsh, Filippo Fregni, Martin Paulus, those who basically help us to be able to run these studies, and all those brilliant PhD and postdoc students. Thank you very much.